Now let us discuss about Good Manufacturing Practices Guide for Active Pharmaceutical Ingredients, Part 9. This comes under Q7 Quality Guideline of ICH. Here we will be discussing about validation. So coming to validation policy, the company's overall policy, intentions and approach to validation, including the validation of production processes, cleaning procedures, analytical methods, in-process control test, computerized systems, and persons responsible for design, review, approval and documentation of each validation phase should be documented. The critical parameters or attributes should normally be identified during the development stage or from historical data, and the ranges necessary for the reproducible operations should be defined. This should include defining the API in terms of its critical product attributes, identifying process parameters that could affect the critical quality attributes of the API, determining the range for each critical process parameter expected to be used during the routine manufacturing and process control. Validation should extend to those operations determined to be critical to the quality and purity of API. Coming to validation documentation, a written validation protocol should be established that specifies how validation of a particular process will be conducted. The protocol should be reviewed and approved by the quality units and other designated units. The validation protocol should specify critical process steps and acceptance criteria as well as the type of validation to be conducted. That is, whether it is retrospective, prospective or concurrent, and the number of process runs. A validation report that cross-references the validation protocol should be prepared, summarizing the results obtained, commenting on any deviations observed, and drawing the appropriate conclusions, including recommending changes to correct deficiencies. Any variation from the validation protocol should be documented with appropriate justification. Coming to qualification. Before starting process validation activities, appropriate qualification of critical equipment and ancillary systems should be completed. Qualification is usually carried out by conducting the following activities individually or combined. Design qualification, documented verification that the proposed design of the facilities, equipment or systems is suitable for the intended purpose. Next is installation qualification, documented verification that the equipment or systems are as installed or modified comply with the approved design, manufacturer's recommendation and or user's requirement. Design qualification, installation qualification, operational qualification, documented verification that the equipment or systems as installed or modified performed as intended throughout the anticipated operating range. Next is performance qualification. Documented verification that equipment and ancillary systems are connected together and can perform effectively and reproducibly based on the approved process method and specification. Now coming to approaches to process validation. Process validation or PV is the documented evidence that process operated within established parameters can perform effectively and reproducibly to produce an intermediate or API meeting its predetermined specification and quality attributes. There are three approaches for validation. Prospective validation is the preferred approach, but there are exceptions where other approaches can be used. These approaches and their applicability are listed below. First, coming to prospective validation. Prospective validation should normally be performed for all API processes. Prospective validation performed on the API process should be completed before the commercial distribution of the final drug product manufactured from the API. Next, concurrent validation can be conducted when data from replicate production runs are unavailable because only a limited number of API batches have been produced. API batches are produced infrequently or API batches are produced by a validated process that has been modified. Prior to the completion of concurrent validation, Batches can be released and used in final drug product for commercial distribution based on thorough monitoring and testing of the API batches. An exception can be made for retrospective validation for well-established processes that have been used without significant changes to API quality due to the changes in the raw material, equipment, system, facilities, or production process. This validation approach may be used where critical quality attributes and critical process parameters have been identified, appropriate in-process acceptance criteria and controls have been established, and there have not been significant process or product failure attributable to causes other than operational error or equipment failure. Operator error or equipment failure unrelated to equipment suitabilities. Impurity profiles have been established for the existing API. If these things are available, then you can go for retrospective validation. Batches selected for retrospective validation should be representative of all batches made during the review period, including any batches that failed to meet the specification, and should be sufficient in number to demonstrate process consistency. Retained samples can be tested to obtain data to retrospectively validate the process. Coming to process validation program, the number of process runs for validation should depend upon the complexity of the process or the magnitude of process change being considered. For prospective and concurrent validation, 3 consecutive successive production batches should be used as a guide. But there may be situations where additional process runs are warranted to prove the consistency of the process. For example, complex API processes or API processes with prolonged completion time. And for retrospective validation, general data from 10 to 30 consecutive batches should be examined to assess process consistency, but fewer batches can be examined if justified. So, for prospective and concurrent validation, 3 consecutive successful production batches should be used. Whereas for retrospective validation, general data from 10 to 30 consecutive batches should be examined to assess the process consistency, but fewer batches can be examined if justified. Critical process parameters should be controlled and monitored during the process validation studies. Process parameters unrelated to quality, such as variables controlled to minimize energy consumption or equipment use, need not be included in the process validation. Process validation should confirm that impurity profile for each API is within the limits specified, and impurity profile should be comparable to or better than historical data. And where applicable, the profile determined during the process development or for batches used for pivotal clinical or toxicological studies. Coming to periodic review of validated systems. Systems and processes should be periodically evaluated to verify that they are still operating in a valid manner. Where no significant changes have been made to the system or process and a quality review confirms that system or process is consistently producing material meeting its specification, there is normally no need for revalidation. Coming to cleaning validation. Cleaning procedure should normally be validated. In general, cleaning validation should be directed to situations or process steps where contamination or carryover of material poses a greatest risk to API quality. For example, in production, it may be unnecessary to validate equipment cleaning procedures where residues are removed by subsequent purification steps. Validation of cleaning procedures should reflect actual equipment usage patterns. If various APIs or intermediates are manufactured in the same equipment and the equipment is cleaned by same process, a representative intermediate or API can be selected for cleaning validation. This selection should be based on the solubility and difficulty of cleaning and that calculation of residual limits based on potency, toxicity, and stability. Cleaning validation protocol should describe the equipment to be cleaned, procedures, materials, acceptable cleaning levels, parameters to be monitored and controlled, analytical methods. The protocol should also indicate the type of samples to be obtained and how they are collected and labeled. The protocol should also indicate the type of samples to be obtained and how they are collected and labeled. Sampling should include swabbing, rinsing, or alternative methods. For example, direct extraction as appropriate to detect both insoluble and soluble residues. Sampling methods used should be capable of qualitatively measuring levels of residues remaining in the equipment surfaces after cleaning. Swab cleaning may be impractical when product contact surfaces are not easily accessible due to the equipment design or process limitations. For example, inner surfaces of hoses, transfer pipes, reactor tanks with small ports, or handling toxic materials and small intricate equipment such as micronizers or microfluidizers. The sampling method should be capable of qualitatively measuring levels of residues remaining on the equipment surfaces after cleaning. And swab sampling may be inappropriate under certain circumstances. When the product examples are product contact surfaces are not easily accessible because of the equipment design or process limitation. For example, inner surfaces of hoses, transfer pipes, reactor tanks, and small ports. Or the second case is handling toxic materials. Even in that case, swab sampling is not applicable. And for small intricate equipment such as micronizers and microfluidizers. For cleaning procedures validation, a validated analytical method having sensitivity to detect the residues or contaminants should be used. And the detection limit for each analytical method should be sufficiently sensitive to detect the established acceptable level of residue or contaminant. And the method's attainable recovery level should be established. Residue limits should be practical, achievable, verifiable, and based on most deleterious residue. Limits can be established based on minimum known pharmacological, toxicological, physiological activity of the API or its most deleterious component. Equipment cleaning or sanitization studies should address the microbial and endotoxin contamination for those processes where there is need to reduce the total microbial count or endotoxins in the API, or other processes such as contamination could be a concern. For example, non-sterile APIs used for the manufacturing of sterile products. If non-sterile APIs are used for the manufacturing of sterile process, in such case, equipment cleaning or sanitization study should address the microbial and endotoxin contamination also. Cleaning procedures should be monitored at appropriate intervals after validation to ensure that procedures are effective when used during routine production. Equipment cleanliness can be monitored by analytical testing and visual examination where feasible. Visual inspection can allow detection of gross contamination concentrated in small areas that could otherwise go undetectable by sampling or analysis. Visual inspection can allow detection of gross contamination concentrated in small areas that could otherwise go undetected by sampling and analysis. So along with cleaning procedures, visual inspection must also be done for the detection of gross contamination. Now coming to validation of analytical procedures. Analytical methods should be validated unless the method employed is included in the relevant pharmacopoeias or other recognized standard reference. The suitability of all testing methods used should nonetheless be verified under actual conditions of use and documented. Methods should be validated to include consideration of characteristics, including within the ICH guidelines on validation of analytical methods. The degree of analytical validation performed should reflect the purpose of analysis, and the stage of API process, production process. Appropriate qualification of analytical equipment should be considered before starting validation of analytical method. Complete record should be maintained at any modification of an validated analytical method. Such record should include the reason for modification and appropriate data to verify that modification produces results that are as accurate and reliable as the established method. Thank you for watching. Please subscribe for further videos on pharmaceutical sciences and other related disciplines.